Hello students, today we are going to discuss a very interesting phenomenon that we call it as DNA replication. DNA replication means self-duplication of DNA molecule. Replication is nothing but a process of duplication. When DNA divides into two, it makes two same copies of each, is called as replication or duplication. If we understand, replica means the copy, exact copy. So replication is nothing but duplication of original DNA into daughter DNAs. Why the replication of DNA requires in the first place? So replication is basically a process which occurs when the cell multiply. For example, we had discussed many times that there is a cell division process in biology. So when cell gets divided into two, one cell gets divided into two cells, at that time what happens, whatever DNA or genetic material which is present in a parent cell, it has to get divided into two daughter cells. It makes exact copy of its DNA, and then that DNA gets divided into two daughter cells. So here it copies the entire DNA strand. So let's see the process. Basically, there are two main ideas about the mechanism of replication. First one, the conservative replication, where what happens, the original DNA helix remains intact and the identical new DNA material is synthesized, right? So in conservation means the first one is conserved, right? And the second one is semi-conservative method, where what happens, the DNA gets unzipped. So you have a DNA molecule, it will get unzipped and new copies will be formed over these DNA strands, right? So this DNA, the original DNA, will work as a template DNA and the newly constructive strands will be seen. So as this diagram shows, the conservative method, in conservative method, the parental DNA is shown with this pink color. And now let's see further what happens. This remains intact, and with some process, somehow what happens, this basically forms its copy. The totally new nucleotides will come and have certain ways so that the same information which is present in the parental DNA is copied into the daughter cells, and further it will divide. So again this will remain intact and the new you can say the DNA will be formed. Now what happens in the semi-conservative method? It is very important and most accepted method, which says that a parental DNA is this, and as I had mentioned, it gets unzipped and there are certain enzymes which basically works over here, and this remains one original strand, another one will be newly synthesized strand, original strand and the newly constructed strand. So and then this will get separated from here, and as you can see there will be two DNAs formed, where you have one original strand and one, the blue one, is new strand or newly constructed strand. The same process will go on further. So this is semi-conservative method, where the half the template, half the DNA is conserved and half is newly constructed. So that this is the most accepted method. Let's see what are various steps that go on in the replication. First one, unzipping of two strands of DNA. The enzyme which is responsible for this is called as DNA helicase enzyme. This is the most important enzyme over here, which basically causes unzipping of DNA. So what will happen basically, this is the DNA strand. So suppose here the origin of replication is there. Origin of replication is a place from where the unzipping starts. So what will happen? The enzyme helicase will come and attacks over here, and these two strands will get unzipped as we open the zip. Right, and now what will happen? The new nucleotides will be there in the environment, and these nucleotides will join over here, and it will form the new DNA. So this function of unzipping of two strands will be done with the help of DNA helicase enzyme. The next one is, as the DNA unzips, the nucleotides are exposed. These nucleotides are exposed and new nucleotides will join here on the basis of complementary base pairing. What is complementary base pairing? We had covered this in the structure of DNA, that A always pair with T and G always pair with C. Adenine will pair with thymine, and guanine will pair with cytosine, right? So they form the new strand according to the complementary bases, joining of complementary bases. Now the new nucleotides, they line up along the DNA template strand. So this is the template strand. The old one will be called as the template strand. Template means it has information in terms of certain nucleotide sequence, and the new nucleotides will come and join in a complementary manner. So like you have in sequence A, T, C, G, G, A, A. So what will happen? The new strand will be formed, and since DNA is forming, A will pair with T, T will pair with A, G, C, C, T and T. So this will be the new strand, and this will be the original strand. So this is how the complementary, this original DNA will function as a template strand, and the new strand will be formed. And this function is performed by a very important enzyme over here that we call it as DNA polymerase. Now, if you can recall, many nucleotide is a single unit of DNA. When many nucleotides, they join together, they form a chain that we call it as polynucleotide, right? So the polynucleotide, this process is called as polymerization. So the enzyme which does this activity over here, we call it as DNA polymerase, because the reaction which is going on is called as polymerization. Next, formation of phosphodiester bond between new nucleotides. That means these nucleotides have come over here, and now who will do the sealing, right? So the joining will be done by the enzyme which we call it as DNA ligase. So it functions as a glue. It functions as a glue. It is responsible to join the nucleotide sequences. So that is why we call it as, it functions as a glue, and its name is DNA ligase. Okay, then this is the example where the diagram where there is a diagrammatic representation of the replication. So we are aware that this is our DNA strand, DNA helix, where these nucleotides are joined with the help of weak hydrogen bonding, isn't it? Now, it results in two molecules of DNA. So, new DNA consists of what? One original strand and one new strand. So, how it occurs? Let's try to understand the process now. See, this is your helix, right? This is your DNA helix. And we are aware that these are 5 prime, one is 5 prime to 3 prime, and another one is anti-parallel to this, so it will be 3 prime to 5 prime. Now look at this, that how it will happen. This is your helix, DNA helix. So suppose here there is, or here there is origin of replication. So origin of replication is nothing but a point from where the process of replication starts. So what happens in case of prokaryotes, there will be single origin of replication, while in eukaryotes, since the size of DNA is huge there, so you have so many replication origins of replication there. So it may form a structure like this, like this, like this. So this is your DNA unbounded, and this forms. Here the helicase will work, helicase will work, and this will open up, the strands will open up. So this forms many bubbles. So they may also call it as replication bubbles. So this will happen in eukaryotes, while in case of prokaryotes, just the helicase will come and join here, and the strands will open up. So this is your helicase enzyme. Unzipping of DNA occurs over here. Two strands get separated. So this is one strand and this is another strand, right? Now what will happen? To begin this process, a very small, minute, a primer is required. So which basically originates this process is called as RNA primer. So this is a primer. Primer is nothing but the complementary bases to the starting nucleotides which are present on the template DNA. So this RNA primer will come and join over here, and after that what will happen, the nucleotides will come and join, like I had mentioned that if you have A, G, G, C, A. So what will happen? The nucleotides are there in the environment. They will come and join. So the complementary base to A is T. Complementary base to G is C. Complementary base is G to C. C to G. A to T. A to T. So this is how the complementary bases will come and join, and they will form a structure like this. So this formation, if you look at this, on one strand where you can see this is your 3 to 5 strand, and this one is 5 to 3 strand, right? So this starts from 5 and 3. So here what will happen, the synthesis of DNA will be from 5 prime to 3 prime. And remember, the DNA polymerase enzyme works accurately when the synthesis is from 5 prime to 3 prime direction. And if it is inverse, that means if it is 3 prime to 5 prime, that means in reverse direction, at that time the RNA polymerase, it doesn't work properly, and so this complete sequence will be not there. You will see small fragments. You will see small fragments over here, like this, like this, like this, and these segments are called as Okazaki fragments. Okazaki fragments, and here comes our most important enzyme, that we call it as DNA ligase, because it joins these fragments together. It does sealing or healing of these fragments, joining of these fragments, and here it forms a complete nucleotide strand. So now what will happen, if you look at this from here, now you have this original strand and you have this newly constructed strand. This is new strand, this is new strand, this is original strand. So now one more thing that you have to keep it in mind over here, that the strand which is constructed continuously in a continuous manner is called as a leading strand. The strand which has constructed, which has synthesized over the template of DNA in a continuous manner. That means which has been synthesized in 5' to 3' direction, which is continuous, right? So that is called as leading strand, while the strand which is formed in the form of fragments, the Okazaki fragments, or in the discontinuous form is called as the lagging strand, right? So we have now leading strand and the lagging strand, where and this one is called as original, and these two are newly constructed. This is again original. So this is how we get the result of DNA replication, where you find one original strand like this, the light blue, and one new strand, the dark blue or gray, whatever it looks like to us, right? Because somehow the resolution changes on the screen, so somehow there is variation in colors. So this seems dark gray to me, so which is newly constructed DNA strand. So this is how the process of replication occurs. Now look at this. These are some vocabulary that we need to discuss. Conservative replication, semi-conservative replication. Conservative, where one whole DNA remains intact. Semi-conservative, one original strand and another one is newly constructed strand, right? And this had been basically done with the experiments where they have used the nitrogen isotopes, like we have N14 and N15. So they have incorporated N15 and they have checked with the experiments that yes, it happens that one strand is original strand and another one is newly constructed strands. DNA helicase, it is an enzyme which is involved in DNA replication and responsible for unzipping of two strands. DNA polymerase is an enzyme which is involved in DNA replication that lines up new nucleotide along the DNA template strand. And DNA ligase is an enzyme which is very important, which basically catalyzes the formation of phosphodiester bond between the nucleotides, and the nucleotides will form a single strand, right? Now, if we will just summarize whatever we have studied, so we have a double helical DNA. The helicase enzyme will come and join here, and that is how what will happen. The unzipping of unwinding or unzipping. So replication fork will be formed, a Y-shaped replication fork will be formed. Here what will happen, we have a primase, so it synthesizes the primer sequence, a small mRNA sequence that we call it as RNA primer, and after that the continuous synthesis of DNA strand, and at another you have fragments will be synthesized. So discontinuous synthesis, that is called as lagging strand, and this is called as leading strand. Synthesis is possible in 5' to 3' direction, so which is 5' to 3' will be called as leading strand, right? And then primers, they are removed, and these primers will be removed, nicks will be sealed, healing will be done, and both of them will be a continuous DNA sequence or DNA strand. Then the proofreading will be done by DNA polymerase. Whatever document we prepare, we just edit it. We do the proofreading, that whether we have left some errors in writing or any documentation. Similarly, this DNA polymerase enzyme does proofreading, and it checks that whether there are any errors in the newly synthesized strand, and then they will get detached from here, and as a result we will get two daughter DNA strands which will be then organized into chromatin structure. And what is chromatin structure? It is supercoiled structure where you have a DNA sequence, and that is basically been held with the histone protein. Histone protein. What is the function of histone protein? It basically, see what happens. Your DNA's length is in meters, and your cell, that to your nucleus, is in the range of certain micrometers. So have you ever imagined that the so long DNA, how it gets accommodated in such a smaller cell, a small nucleus? So why? Because it is a supercoiled structure. It is very much supercoiled, and there are certain proteins that we call it as histone proteins, which basically bind these DNA threads, right? And that is how here duplication occurs, and now these two DNAs are ready to get separated and to enter into the new progeny of the living cell. So that is how we have seen today replication. It's a very interesting method, and I would say that I have explained it in a very interesting way with the help of diagrams. So stay tuned for many such topics. Thank you so much.